biotechnologies work in a similar way but are less controversial. They include genomics, molecular-assisted selections, and the genetic engineering of crops. These allow researchers to detect and map genes, discover their functions, and select the specific genes in genetic resources in breeding, and transfer genes for specific traits into plants where they are needed. NIFA funds research, training, and extension for developing and using biotechnologies for food and agriculture. Areas of work include genetic structure and mechanisms, methods for transgenic biotechnology, also known as genetic engineering, and bioengineering, identification of traits and genes that can contribute to national and global goals for agriculture, plant genome sequences, molecular markers, and bioinformatics. Oh my God!
Hello and welcome to That's Humorous. I'm your host, Judy Arnold, and I'm here to welcome the renowned Alfred Sternman. Now, Alfred, could you give a little insight into why you're here today?
Back when I was an undergraduate researcher in the laboratory of Thomas Hunt Morgan, I realized that I had to map the locations of the fruit fly genes whose mutations the Morgan laboratory was tracking over generations to order my data. This led to the creation of the Human Genome Project.
For those who are a little slow in the audience, could you give a little explanation as to what the Human Genome Project is?
The Human Genome Project was the international collaborative research program whose goal was to complete the mapping and understanding of all the genes in human beings. All of our genes together are known as our genome.
Sternman's first mapping of the genomes can be likened to the Wright brothers' first flight at Kitty Hawk. In turn, the Human Genome Project can be compared to Apollo bringing humanity to the moon.
Yes, the process is quite intense. So the hereditary material of all multicellular organisms is the famous double helix of DNA, which contains all of our genes. DNA, in turn, is made up of 4 chemical bases, pairs of which form the rungs of the twisted ladder-shaped DNA molecules. And all genes are made up of stretches of these 4 bands arranged in different ways and in different lengths.
How did the researchers decipher the human genome?
It's done in 3 major ways, determining the order or sequence of all our bases in our genome's DNA, making maps that show the locations of genes for major sections of all our chromosomes, and producing what are called linkage maps, which are complex versions of the type of originated in early research, through which inherited traits, such as those for genetic disease, can be tracked over generations.
We'll be back to this riveting topic in just a moment. Marine biotechnology is the study of how the various organisms and actions of the ocean can be used to provide services and products to us. There are 3 main challenges. The first challenge, biosafety. Testing living modified organisms may disrupt the natural ecosystem of the ocean. The second one, access to marine organisms and resources. Because marine biotechnology takes place in the ocean, the borders between different nations can sometimes erupt in disputes over access to certain resources and creatures. And lastly, the third, intellectual property rights. The North wants stricter rights to discoveries made in the South, where marine resources are high, while the South feels that because the North is researching in their property, they should be able to reap some benefits. Many new solutions are provided to industry and agriculture, including environmentally friendly pesticides and salt-resistant enzymes that are helpful in many industrial applications. Welcome back to That's Humorous! That's my line. Let's try this again. Welcome back to That's Humorous. I'm your host, Judy Arnold, and we're here with Alfred Sternman to talk about some questions from the audience.
How many genes are there?
There are approximately 20,500 human genes, and the complete human sequence can now identify their locations. This ultimate product of the HGP has given the world a resource of detailed information about the structure, organization, and function of the complete set of human genes. This information can be thought of as the basic set of inheritable instructions for the development and function of a human being.
How do they construct the first draft? Could you refine it?
Techniques include DNA sequencing, the employment of restriction fragment length, polymorphism, yeast artificial chromosomes, bacterial artificial chromosomes, the polymerase chain reaction, and electrophoresis.
What did one DNA say to the other?
What?
Do these genes make my butt look big?
And on that note, we're off. Stay tuned for our next special guest.
Have you or a loved one ever been the victims of a homicide? Have you wanted to find your murderer? Now there's no longer the need to rise from the dead and haunt your killer. With my 6-stage guide to success, you and your loved ones will finally get the peace of mind you've been waiting for. Starting with the alternate light source that we provide to find blood and semen. Was there a blood splatter? Use our blood stain pattern analysis kit using luminol for testing for human blood, or phenolphthalein, also included in the kit. Was there semen on the scene? Use the chemical analysis BCIP to identify it, also in the kit. Were there any fingerprints on the scene or latent printed evidence? If so, choose from our many tools included in the kit. Amido black, fingerprint powder, leucocrystal violet, cyanacrylate superglue fuming, and titanium dioxide, electrophoresis dust lifter, dental stone casting material, and sulfur casting. Was there any trace evidence, such as firearm and tool mark evidence? Pick one of the following to further identify your subject. Gunshot residue, trace metal detection, sodium rhodozonate test for lead, silicone-based casting materials. Also included as a bonus, we will provide general equipment used in processing scenes, such as metal detector, laser range meter, and electronic distance measure EDM, device and data recorder. You can get all of these tools, plus the very high-tech materials that are not shown here, for the inexpensive cost of $999.99 per month for 24 months. If you act now, we'll throw in the question document evidence guide kit for free!
I'd like to give a warm welcome to our special guest, Precious Plum.
Hi, y'all.
Where are you from, little girl?
You.
What?
We have a special surprise today. It's Paternity Test Tuesday, and we're about to play a round of Who's Your Daddy?
What's going on here?
Precious is here today to shed a little light on a special subject. We have to do some genetic testing to find out if Miss Plum is your daughter.
What?
Luckily for us, Precious is a jack of all trades. She'll be conducting the test herself. We'll meet you back in the lab.
What?
To start the forensics biotechnology process, we first took the DNA of my suspected father and I. This was done by taking cheek swabs. Then I moved on to the lab. I dyed the DNA for the cheeks so you could see them easier. And I worked with the DNA to show you how the process works. I mushed up my cheek swabs for a minute or so to make sure it was not chunky. Then I added 10 milliliters of buffer solution to remove the nuclear envelope surrounding the DNA. Once the two were mixed, I added it to a test tube using a cheesecloth, so no chunks of skin were added to the tube. Although not shown in the video, from there I filled the test tube with cold alcohol so that the DNA could be seen twisted up the tube. Moving on to the processing the DNA, I micropipetted both him and my DNA into a agar gel mold surrounded by a buffer solution. Then I hooked it up to an electrophoresis chamber to let the DNA run, which takes some time. Once it finished, you can see the results.
We're back, and the results are in. Congratulations, it's a match!
What the failure?
Actually, it passed. Science in the kitchen for your 5-minute entertainment between shows. Today we are going to make cheesy mushroom bread. This is a perfect example of microbial biotechnology, whose research is critical in the advances in food safety, biotechnology, value-added products, human nutrition and functional foods, plant and animal protection, and furthering fundamental research in the agricultural science. Microbes are organisms that are too small to be seen by the unaided eye. They include bacteria, fungus, protists, microalgae, and viruses. I'm your host, and today I have 2 guests. The first one, don't mind her. She's a bit cheesy. And the second one, don't mind him. He has a dry sense of humor. Did you all bring your ingredients today?
Yes.
Who brought the bread?
I got the bread.
Bread is a great example of the use of yeast, which is a form of fungi. Who has the cheese?
I've got the cheese.
Did you know that's a great form of bacteria? And I've got the fungus, which is shrooms today. Yeah.
Okay, so we're gonna make some cheesy mushroom bread, right?
Yep, that's what I heard. So, due to budget cuts, we're using a paper plate. So you first slab on that good old healthy bread.
The lukewarm cheese.
Best if it's processed. And then you put on those good fungus. Then you stick it in the microwave. Would you do the
For
honors?
exactly 1 minute and 30 seconds.
So cook it just until your cheese starts boiling and it is smelling great. So we can all take a bite. Yep.
Yeah.
You go, bread.
Yes.
It's all for you. Take a good old heaping bite. You actually bite down.
I'm not biting
I think that's
down.
good. That was perfect.
Kind of.
Go.
Amido Black. Fingerprint powder.
Okay. And then just...
Welcome to Science in the Kitchen for your 5-minute.
For your 5 minutes.
Is this fun?
Maybe drool a little.
No.
Hold on, I'm not in the right position. Okay.
The laptop's in this shot. Okay.
It was going good.
Okay. Ready? 1, 2, 3.
Of human genes. This information can be thought of.
I'm your host, and today I have 2 guests. What the heck?
Smile. Oh.
Where?
Read it. There are approximately 20,500.
Okay.
Identify their location. What are you doing?
Hold on. I want to.
I have a question.